How is the cell cycle controlled? If the cell were to proceed from one phase of the cycle to the next without assurance that all the duties of the previous phase were completed properly, serious mistakes might occur at cell division. To avoid such mistakes, assessments must be made at specific moments during the cell cycle, called cell cycle checkpoints. The first checkpoint occurs late in the G1 phase. At that time, the cell cycle will be arrested until the cell size is adequate, nutrient availability is sufficient, and certain growth factors in the form of signals from other cells are present. When growth factors stimulate a cell, they trigger the cell to increase its concentration of cyclin. The G1 cyclin is a protein that binds to and activates the enzyme cyclin-dependent kinase, or CDK. CDK, in turn, activates proteins needed for the S phase to proceed. For the cell to pass through the G1 checkpoint, this cyclin-CDK complex must be activated. Once the G1 checkpoint has been passed, the cell can then enter the S phase. At this point, G1 cyclin is degraded, so CDK becomes inactive again. There is no checkpoint for passage to the G2 phase. During the G2 phase, the cell must commit itself to mitosis. The second checkpoint occurs at the end of this phase. At that time, the cell cycle will be arrested unless cell size is adequate and chromosome replication has been completed successfully. As at the G1 checkpoint, progression beyond the G2 checkpoint involves activation of CDK by G2 cyclin. This cyclin-CDK complex is named mitosis promoting factor, or MPF. MPF initiates mitosis, which proceeds up to the point at which the separated sister chromatids move toward the poles of the cell. A final checkpoint occurs during metaphase of mitosis. At that stage, the cell cycle will be arrested unless all chromosomes are appropriately attached to the mitotic spindles. When the metaphase checkpoint has been passed, mitosis is then completed as mitotic cyclin is degraded. The cell cycle then repeats itself, creating additional generations of cells. The CDK molecules persist with periodic activation at checkpoints as the corresponding cyclins are synthesized and then degraded.